whatever cultural assets that you have, how to become modern. With you don't have to leave. Okay, we are live.
Okay, that's really nice of you. Great. So thank you so much. Thank you so much for joining today's event with Dr. Nandini Verma. And I would like to start with saying good morning to everybody, to our participants, to our speaker, Dr. Nandini Verma today. And welcome to SciComm Synapse interactive talk today and Sunday morning.
Thank you very much.
And not without wasting much time, focusing more on the event. We should start with the event itself. So I would like to request my teammate Bhakti Sadguru to please kindly introduce Dr. Nandini Verma today.
Good morning, everyone. Let me quickly introduce our today's speaker, Dr. Nandini Verma. She is an esteemed principal investigator at the Advanced Center for Treatment Research and Education in Cancer, where she leads groundbreaking research in the field of cancer biology. Dr. Verma's academic journey is a testament to her dedication and excellence in the sciences. She completed her bachelor's degree in biotechnology from Gargi College, University of Delhi, followed by a master's degree in molecular biology from Delhi University. She then pursued her PhD in cancer genetics at CSIR Institute of Genomics and Integrative Biology, where her doctoral research laid the foundation for her lifelong commitment to addressing the complexities of cancer. Doctor Verma has been the recipient of numerous accolades for her contributions to science, such as she served as an assistant professor at Homi Bhabha National Institute, Mumbai. She also got prestigious Koshland Fellowship by Koshland Foundation, McDonald Liebman Grant, and Senior Postdoctoral Fellowship for Academic Excellence at Feinberg Graduate School, Wisman Institute of Science. Her work has been recognized both nationally and internationally, reflecting her impact on the global research community. Her research interests encompass the molecular mechanisms underlying cancer progression, drug resistance, and tumor microenvironment interactions. She is particularly focused on translating fundamental findings into therapeutic interventions, bridging the gap between bench and bedside. Dr. Verma's dedication to advancing cancer research, coupled with her leadership at ACTREC, continues to inspire a new generation of scientists and contribute significantly to the fight against one of the most challenging diseases of our time. Ma'am, we are very honored to have you today as a speaker. And with this warm welcome, I now request the host to continue the event. Thank you so much.
Thank you so much, Bhakti, for the introduction. And I first want to congratulate all of you because you have taken something which is very necessary for young students. Unless people know and get inspired that biology is very good subject itself, research is a very nice subject, and people can really do that, right? So people will not be inspired. They will not take up this as a career, and we need more and more people pouring into these kind of efforts to help mankind. So all of you are doing fantastic job. I really congratulate all of you, and
Thank you
thank
so
you
much,
for
ma'am.
inviting me here.
Thank you so much, ma'am. That was a really nice. And thanks for the kind introduction, Bhakti. So, ma'am, when we see today's title of the event, chasing the cure, drug tolerance, and targeted therapeutics in breast cancer, and since you are working in this field for quite a long time, like if somebody asks, then in layman language, or maybe for an undergrad, what does drug tolerance itself mean?
So I would say that it is a concept for solid tumors most of the time. People are
Mm-hmm,
now
mm-hmm,
starting to see the same concept in other tumors also. But what
mm-hmm,
happens, you can imagine that solid tumor is a ball of multiple type of cells. Most of the time they are having tumor tissues into that, and when a drug arrives there, you have a gradient of everything actually within the tumor.
mm-hmm,
The tumor
mm-hmm,
core that has less oxygen, the drug reaches
mm-hmm,
less there. The reprogramming
mm-hmm,
of mRNA expressions and metabolism, proteomics, and everything is different in that multiple type of cells which are composing that tumor whole thing. And not only tumor cells, there are also stromal cells associated with the tumor.
Mhm.
And as they
Mhm.
get a drug, not every cell would respond to the same level, right? They will not die or become sensitive to the same drug as other cell in the periphery, or maybe it's in the middle, or maybe in the necrotic area. So
Mhm.
they somehow try. And cancer cell, I would say, they are the smartest. Even they are much smarter than us, and they know how to survive in any given condition, right? So they
Mhm.
immediately they get a time to reprogram, and these kind of reprogramming are most of the time mRNA driven, expression driven,
Mhm.
right? It's
Mhm.
translation, transcriptome driven. So they
Mhm.
reprogram themselves in a way, and maybe several other factors facilitate that reprogramming, which are actually maybe surrounding factors, not maybe the intrinsic
Mhm.
factor of the cells. And with the same genetic background, they could do that. So they adapt to certain state of cell where the drug cannot affect them. So they, even the high amount of drugs are there around the tumor, they are tolerant. Means they somehow don't
Mhm.
bother about the drug because they reprogram themselves in a way that they are not anymore affected by that. And not
Mhm.
many tumor cells can do that. So this happens up to maybe 10 to 15 percent of the
Mhm.
tumor cells. They could do that, and no matter how long you treat them, they still become tolerant. And one of the main thing that people have found, that they become dormant, they don't divide.
Mhm.
So most
Mhm.
of the therapies are targeting pathways which are actually supporting cell division. So if
Mhm.
they
Mhm.
decide not to divide at that scenario, become dormant like a bacteria,
Mhm.
right? So
Mhm.
they will
Mhm.
survive this phase and somehow try to metabolically adapt to the scenario, that they will somehow survive this phase. And once the drug is
Mhm.
off, they start to again divide, and then this is the pool of the cells that actually initiate recurrence of the tumor and also metastasis, because they become very aggressive during that time. So
Mhm.
this happens in many uh, type of cells in solid tumors, and people have recognized in lung cancer a lot, carcinomas of solid, and little bit in breast tumors.
Mhm. Mhm. I see. So, so basically, they also like since you said they remain dormant. So, are they also uh, is the drug can permeabilize the plasma membrane? No, right? Cannot
It
reach,
can,
cannot
it
target.
can, there may be multiple mechanisms to do that,
Mm-hmm. Mm-hmm. Mm-hmm.
and one of them could be that the drug is no more effective or may be thrown out of
Mm-hmm.
the cells. But these are known mechanisms, and this happens for resistance, not for tolerance.
Mm-hmm.
So there's a
Ah,
thin
okay.
line between resistance and tolerance. Resistance can take time to happen. Tolerance can be
Mm-hmm.
reprogrammed within minutes.
Ah, yeah. That's something everybody should get to know today. Good. So since we started with the title and a little bit introduction, I would like to ask you the first question for today's event that Dr. Verma, what made you decide to study science at the first place? Like when did you decide that yeah, you want to pursue a PhD or you want to go ahead in research?
So, to be true, when we were like young in our schools, at that time, I always wanted to know more beyond what people know. This was most of the time my intuitive force, that what is
Mm-hmm.
happening, how it is happening. When I was in school, I used to love astrology, astrophysics, and these kind of things, that how stars work and where's the end of the universe. Because at that time, you live in a fancy world, right? But as you grow, you can see that world is a different, they have different kind of approaches, they know. So I decided to take biology. I used to love physics a lot, but somehow, because I did not like maths a lot, I took biology, because there mechanisms, how things are happening, and these are things that fascinated me at the level of 12th class, maybe 11th and 12th.
Mm-hmm.
Then I want, of course, like many others, I wanted to go for medicine. but
Mm.
somehow it was like maybe the guidance not proper, and maybe multiple factors that affected. And I was quite passionate to go to medicine, but somehow I couldn't do that. And then I took zoology honors in Delhi University, and because I like to study physiology, biochemistry, cell biology, this was my passion to study. And the fact of the matter, like, I was quite happy with that. The same books that we are reading in our BSc level, also the medicine graduates are reading.
Yeah.
So I thought,
Mm.
okay, doesn't matter, maybe, but I am gaining the same knowledge. So I
Mm.
used to like many subjects there, like histology, uh, cytology, and how the tissues are processed for multiple things. Immunology was one of
Mm.
my favorite subject. So BSc was something that made a very good background for us, and understand that
Mm.
what is biology itself and what are the different streams of subjects. But then when we entered in our master's program, it was in zoology department in Delhi University, and I
Mhm.
specialized into cell and molecular biology. This was
Mhm. Mhm.
that time actually, and most of the thanks goes to our professors at that time. We had a
Mm.
fantastic set of professors, and in the second year, even in the first year, they used to teach in a way, not in a bookish way. They used to tell us how things were discovered, how people know with what kind of experiments people could know this fact that translation, this is how translation is happening, how ribosomes are functioning, how cell is actually taking care of the proteins which are no more needed, how people actually investigated with so much preliminary set of equipments at that time, and people used
Mm.
to make serendipities. So that actually made us so fascinated that, see, this can be done. And if we understand enough how to do this, if the equipments are available, if the techniques are available, so people can actually ask questions. And not only those people like Roger Finberg, not only those people, we can also do that, the same thing. That means it's
Mm.
possible to know how
Mm.
things are working. So at this time,
Mm.
we had very fantastic set of professor like Murlidhar sir in biochemistry department. We used to call him KMD sir. Madan Mohan Chaturvedi, he came from BHU, fantastic teacher. And
Mhm.
also Ruplal sir in
Mm.
biotechnology. So there are several more cell biology that we actually got the cream out of what we could get at that time, and that inspired
Mm.
us so much in a way that science can be done even by we people. It's not like this is
Mm.
something like even European people or American people can do. We can also
Mm.
do same
Mm.
things provided we
Yeah.
know enough. So that
Yeah.
was the time. Then I already gave CSR net in my first year end, and also on
Mhm.
the next time. So we used to read a lot, and then within that I got the net exam qualified within my first year of masters, and then I decided, yes, I
Mhm.
want to pursue that because this is something I would
Mm.
love to do, and I could see that I can't do anything else. Of course, I can
Mm.
teach, but I would rather go for investigation more than teaching.
Mm. True. True. That's really true. So now, since you mentioned about your bachelor's and master's time, how struggles and how the way were put during that time. So, like when you entered your PhD, like what was your mindset that okay, like what I want to do in next 4 or 5 years?
So. I was kind of a person that would like to be independent in order to
Hmm.
have my thought process open and to read by myself and to
Hmm.
understand what would happen. So initially, when we were little bit naive at that time, I started my PhD at 2004. The first PhD I joined, it was in zoology department itself. There was some new
Hmm.
professor that came in, and she joined, and she was using developmental biology. Dictostelium was the model. So we joined that lab, and it was new. One, me and my classmate, MSc classmate, we joined together, and because this was something very going very slow, and I was not able to catch up with the system, and this was a new lab. So, and it was a university system. You know how in India university works. So I all the time felt I worked there for almost 8 months for this PhD program. I always felt there's something missing, something which is not my cup of tea. It's maybe very slow. Things are not happening on the pace. I
Mm.
always wanted a dynamic culture and more people to talk about what you are doing. So somehow I was not sure that I want to continue this lab or this subject per se. And at the same time, my professor actually wanted to move to JNU. So I was thinking, okay, then maybe now it's a high time I should decide and I should change my, stream
Mhm.
or also the subject. So then I entered to CSR Institute IGIB after that time.
Mm.
I decided to give up, and at that time, nothing was there in my hand. But I decided,
Mm.
yes, this is not what I want to pursue. This is not what I can love. I need a different type of culture, different type of question.
Mhm.
So I entered IGIB, CSR Institute at Mall Road in Delhi, and there I tried to explore what I would like to do. And I met Dr. R.H. Das, Hari Das. He was very senior professor at that time at IGIB, and he used to work on multiple things like baculovirus system, some
Mhm.
plant-based enzymes. So he was versatile person. He could take any kind of
Mhm.
question with his experience and could make your way. And I was very much open to what you want to do. It's not like he
Mhm.
would impose on you what you have to do. He always asks a
Mhm.
question. Okay, you are bright, you are intelligent, you have your own fellowship. What is something that is intriguing you? What kind of question you would like to ask? So I explored, and then I came to know that there are some setup in this lab where people are also addressing some innate inflammation, innate
Mhm.
immunity in macrophage
Mhm.
culture. So I thought maybe this is something I can pursue. And
Mhm.
then I said yes to Dr. Das, and then we started this PhD thing. And initially, I was working on some bacterial system also, so I learned a lot HPLC, many techniques I learned in the beginning.
Mhm.
And then
Mhm.
he in one day said, "No, okay, you have done whatever you are doing, but now is the time to decide your question." So that
Mhm.
time he said, with all set of techniques and what you can do, I would suggest you to go for a complex kind of system where you can, discover something that you can actually do something. And this is something
Mhm.
innate immunity and this question with how macrophages are working, how TNF receptors are working, how they are bringing about the death in the, activated macrophages. This is something that you can pursue with certain
Mhm.
receptors and
Mhm.
enzymes which are important for the innate immunity. So that is
Yeah.
something
Yeah.
I really liked, and I took this topic up, and then I started my actual work that I had to do. So in the beginning, I struggled a bit and explored
Mhm.
in different university level, and the fact was like at that time, when I entered IGIB, I could sense the complete difference in the culture, the institutional
Mhm.
culture and the culture in the universities. Even DU was
Yes.
very good in universities, not like that it's
Yeah.
not competent enough, but the environment and the culture and the dynamics, and people
Mhm.
are talking about their science, they're helping each other, they are sharing the reagents, they are activating each other, what you are doing, we have done there. So that was something which really I loved, and I learned that culture from there. And that was something that I was like in love with science, that I wanted to pursue for till my end of my career. I would be
Mhm.
like I would like to do that. This was something.
Mhm.
So this
So
is how I did what I did in my PhD.
So the priorities really like matter in PhD. You need to see what you want
Yes,
and what you really are looking for.
right.
I think.
You must settle down with something that you really want to do. If you will not do
Mm,
that, probably this is not you are doing the PhD. Somebody else is doing your
mm,
PhD.
True that, true that, true that. So, since you mentioned about like joining IGIBs from DU, then like what were the major hurdles or the like very small issues before, and which like probably students don't face nowadays? So with so what do you think is the in the difference?
Uh, I think at that time there were some things which were very slow, like procurement of reagents, which has actually changed a
Mm,
lot. But still, it's not too much in India, I would say. This is one thing we
mm.
used to wait up for our reagent like 45 days, 1 month.
All right.
For some things, it still have not changed, even after so many years.
Mm-hmm.
Another hurdle that maybe not me, but many people face, that the availability of their mentor for discussions.
Mm, mm,
right? Many people are so busy, and that thing goes to the, like, lot of administrative jobs has been assigned to PIs. They get very less
mm,
time
mm,
to actually do science. So because of that, they
mm,
are busy in multiple things which are maybe not necessary. And this is the culture that I see, that outside India, people are more focused, and they could focus more onto their research topic, and they can, have collaboration, write more grants. But somehow our system is less, competent to allow a scientist to read, write, mentor, talk about science.
mm,
We are
mm.
more busy in multiple administrative jobs and pushing other people.
That's certainly right. So now, in your PhD days, like what is the one thing that you would remember forever from your PhD? Like if any experiment or maybe something with your mentor or a paper.
There are so many things actually that I would say, but some of them which are like, when I was making some sepsis model in mice. Uh,
Mm-hmm,
why I was doing that? Because I have seen that when people enter, I was working on an enzyme called iNOS, which is inducible
mm-hmm,
NOS.
mm,
So I wanted to ask a question very relevant, that how this enzyme is important in a physiological way. And I came to know that iNOS actually is highly increased when people go sepsis, and this, because of this, the blood pressure is actually down and people die because of cardiac arrest. And this happens in multiple, people who are admitted to ICUs, and eventually they get infection. iNOS is increased, their blood pressure drop, multi organ failure happens, and eventually people collapse out of cardiac arrest. So I wanted to understand that maybe this is most important question when we talk about INOs. And I made a sepsis model in mice. And when I injected antagonist shRNAs and other RNAs to INOs, and we could see that the mice are surviving, even the lethal dose of LPS that can induce sepsis into them. So that was something I couldn't believe to that level. Ki, my God, like, it is possible. At least in mice,
mm,
when we are saying that half all the mice in the control cohort are dying, and we can save the other mice just because
mm,
you are able to inhibit inos. So
mm,
see,
mm,
this is
mm,
a matter of life, and that this is something I remember, that yes, and my supervisor was so happy, and he went with me to the animal room, and he said, saw everything, and he then helped me that okay, we must do histology of all these organs, what happened.
mm,
So then
mhm,
I went to
mhm,
Safdarjung Hospital with all the organs in the tube fixed and did the histology. So this
mhm,
was something which is something called seeing is believing, right? And when you see
mm,
such thing,
mm,
you see, yes, things are possible, but you just need to do it
mm,
right. And with your mentor, they are pushing you when they could see good results. So he was super happy.
mm,
Okay, oh, something happened, really
mm.
happened. When you look at cellular level, okay, cell can die, cell can survive, but that's not physiology, right? People
Yeah,
need to look at
yeah,
the systemic
true.
level, what is the overall effect on the survival. So
Mm,
that was one
mm,
moment that
mm.
made me so happy, so content, that yes, you are somewhere right. You're doing, and things are working. There is
So
science.
correct. There is science. So
Yeah,
basically, correct mentorship and
yeah,
with your intelligence, you can really come out with exciting projects. That's definitely sure. So this was something that was like a light bulb moment
right.
for you. That's really nice. So after, like, since you were now like ahead of your PhD, like when did you decide, or was it pre-decided that you would go for a postdoc abroad, or like, yeah?
Yeah, it was automatic. One thing, because
Uh-huh.
of the culture that we had in IGIB, that
Mm-hmm.
almost 85 or maybe 90% people choose to go for higher studies and for postdocs,
Mm,
because that was the
mm,
culture that already pushes you towards that. There's no other settlement. You
mm,
have to pursue much higher.
mm,
You need to become more intellectual, do better
mm,
science. So most of our
mm,
people in IGIB used to go for postdocs, and that also something
mm,
that also helped us to decide. But yes, nevertheless, I always wanted to go for postdoc. That was not even a question that I would not go. That was
mm,
straight
mm,
away. I knew that I have to, and I want to become an independent scientist later on after
mhm,
my experience in
mhm,
postdoc. It was automatic and
mm,
probably very
mm.
natural to me.
So when you like landed in a different country, like what was the first thing that you thought, like leaving everything behind, like coming for a postdoc?
To be true, I used to be very strong person in general, that I could
Mm,
give up whatever for my career, or whatever
mm,
can come in
mm,
my way. I will
mm,
not treat it as an obstacle. I will just eliminate that. But
mm-hmm.
when and I always wanted to go US, but somehow that time
Uh-huh.
recession was there, and whatever lab I wanted to go, one lab I could, but then funding was an issue and so on. So
Mm,
I
mm,
was looking for some project that where I can really apply what I want to do and what I know, and
mm,
what I can handle.
mm,
So
mm, mm,
suddenly, like I thought, okay, somebody, one of my scientists in IJB told me, oh, Weizmann is also a very great place. It's one of the
mm,
top 10 university in centers in
mm,
science.
mm,
Then I thought, okay, let's try to look there. So
mm,
I first applied to one professor, which was working on immunology itself, and we used to read a lot of papers of him.
mm,
So then I applied to him. He said he took little bit time to. He was interviewing me. In the meantime, I also wrote some other professor also.
mm,
So one
mm,
project was on breast cancer,
mm,
finding out the role of some focal adhesion kinase. And
mm,
I thought, okay, this is also an important question. So I also write to that professor also. So luckily, my mentor in postdoc, she replied faster than the other one, and
mm,
then I said yes to her. And the
mm,
other one that was in my field in immunology, he took time and he said, "Okay, I also like you, and I would like to welcome you."
mhm,
But I said, "Sir,
mm,
but
mm,
something happened in between," and I said yes to other project in the same institute itself. And I
mm,
told the
mm,
name of that person, and he said, "Okay, yes, that's correct. She's also a very fantastic scientist, a very high level
mm,
scientist. You should go
mm,
ahead." So then I decided, and when I landed there, it was kind of a cultural shock. The first thing I saw when I went to Rokovot, which is where the Weizmann is, that very young boys and girls. That was Friday or some other day when
mm,
I landed. Many people are
mhm.
roaming on the street with guns because they did army training, right?
Yeah,
So after 12th, they entered to their
I
army
mean,
training for 2 years and 3 years. So I was like, oh
yeah.
my god. And the second thing, like they read and write Hebrew. In so
Mm.
when I went to market to buy some grocery, I could not read any label there. I was like, oh my god, I'm completely, illiterate kind of thing.
Yeah.
As soon as you entered, and people are just, shopkeepers are already, they don't understand English enough. So there was an initial challenge, and the culture and everything was quite different. In some way, it is also
Mhm.
similar to India, but it was dramatically different. And you were
Different,
you were
yeah.
expecting that okay, it would be something like US cosmopolitan, but it was not like that. But yes,
Mm.
I would say people were very helpful, especially the Weizmann Institute foreign student unit, that really helped us
Mhm.
a lot. And then did not
Mm.
let us feel this way, that okay, you are alone, you are stuck somewhere, you won't be able
Mm.
to manage. So everything was very
Mm.
smooth. The only thing that I could little bit learn Hebrew in the all the time that I was there. But
Mhm.
overall, it was a very good experience. But yes, initially, maybe I would say 1 or 2 months was very tough. And at that time, Google Translate came. So we used to take photos of the things and translate, and then could buy anything or something which is a transparent level where we could see that, yes, this is
Mm.
some
Mm.
dal or some rice. So this
Mm.
was a
Mm.
struggle in the beginning, but we cope up very quickly with that.
That's so you were the meta tester for the Google Translator that
Right,
we are
right. It was
using.
just came at that time. So we used it
Mm.
like anything.
Mm-hmm. Mm-hmm. So, what was your like project for your postdoc? Like, I understand you would have done several projects, but like maybe one key highlight of your postdoc, which could be a career changer or something like that for you.
Uh, Actually, I was looking. There was one tyrosine kinase called PYK2, which is a sister family of FAC family. So
Mm.
I was trying to look that what is its role, and I was looking probably in a wrong cell line. which is
mm,
luminal type of breast cancer. So breast cancer have very much many subtypes. Some are some
mhm,
are luminal, which originate from lumen, and some
mm,
are basal in nature. So
mm,
I was
mm,
trying to do experiments because the cell lines were actually available at that time. So I
mm,
was trying to
mm,
look whether it is phosphorylated, not phosphorylated, what is happening, IP experiments.
mhm,
I took almost 5 months to figure out what is happening with this PYK2 molecule, but I was not successful. IP after IP, thousands of IP, growth factors, this, that. Then I decided, no, something is wrong again. It cannot be like that. If the cell is expressing so much of this enzyme, there must be some function, right? Then
mm,
I went to some literature and find out where this enzyme is highly phosphorylated, this tyrosine kinase. And then I found the set of breast cancer where it is highly activated, highly phosphorylated is not luminal but triple negative breast cancers. So that was the day I decided I would like to work in triple negative breast cancer,
triple
taking this tyrosine kinase as my molecule.
negative.
So there I
Mm,
started to read, understand. Then I realized this is a very bad type of cancer, a very aggressive one. I must work on
mm,
that. So that was the molecule with which I entered into that field, and that was, I still remember the day. It was 2007, 12, 11th of September, where I got this blot that picked is highly phosphorylated in these set of cells
mm,
in EMT conditions, highly activated, highly phosphorylated. And I still
mhm,
remember that
mhm,
plot, and I was super happy that it is activated there. And since then, I did not stop back and look back.
mm,
And then I took this enzyme for two more projects, and then I did other type of
mm,
things. But then I entered into the field of triple negative breast cancer. I collected all cell
mm,
line of triple negative with
mm,
my mentor. We worked in almost 12 cell line of different subtypes and so on. So that was something
mm,
that made me decide to see, when you try to look in with the wrong thing, you will not
mm.
be successful. You need to really
Yeah.
understand why it is happening. And once you understand,
Mm,
there's no stoppage. You just have to work and
mm,
everything works for you.
mm, mm, mm. That's really
And then even I
that's really
discovered
true.
that this enzyme, which is supposed to be a focal adhesion kinase, supposed to be in the focal adhesion, it is moving
Mm,
to the endosomes with the EGFR receptor. And that was a discovery,
mm,
nobody could show it before, that it is actually
mm,
translocating with EGFR to endosomes, and there actually it is protecting EGFR from the degradation. So that was something when
mm,
I looked into confocal and said, oh, why this enzyme is moving somewhere, it is leaving
mm,
focal
mm,
adhesion, why? So then we investigated
mm.
further and could see that.
That's so you need one idea in your
Correct.
mind
Yeah.
that
Right.
could completely change
Right.
the stars. That's really true. So like since you moved to Israel, what difference did you find in the mentorship or in the lab setting, that
There's a
helped
lot of
you or
difference. Yeah.
mm-hmm,
First of all the setup in that kind of institute is like pro research.
mm,
Everybody there will facilitate you. This is one thing that
yeah,
I would say. And you don't
yeah,
need to roam around for multiple things. You just drop an email. System was
yeah,
so smoothened to facilitate you as a researcher. This is one thing. Another thing, most of the people in Israel Weizmann Institute, uh, they are people who have done a groundbreaking discoveries in their field. Groundbreaking means they were pioneer in something. One of the professor whom I applied, he was a discoverer of TNF receptors, right? So somebody actually discovered stem cell for the first time. Somebody
mm,
was like, these people are actually a collective, you can say a beautiful basket of scientists who did
mm,
groundbreaking discoveries in their field. Really
mm,
groundbreaking. My
mm,
mentor actually discovered this PYK2 kinase and multiple other
mm,
kinases. And so
mm,
these were people who were discoverers of something.
mm, mm,
Other people, they discovered P53 gene, right? So
mm.
these were the people around us. So when you see
With
such people, such scientists who really made changes in this world, how people know about things, you feel
mm,
so fantastic. You feel so ignited. You feel like, oh my God, we are among those people who did such important
yeah,
discoveries in this field. So
yeah,
you are at a different level of thinking
mm,
process, of execution, of not asking redundant questions. You need to ask a very
mm,
important question. This is something I
true.
learned from there. What type of
Mm,
question you should be asking? What makes
mm,
difference in the field?
mm,
What would actually facilitate other things? So this
mm,
is very important to ask a good question and already think about what it might do if you discover something. To be a
mm,
visionary. So most of the scientists around us in our buildings and wherever we go and had a talk, we felt like, oh my god, we are into a different system and we can do a lot.
mm, mm. So asking the correct question makes a difference as well.
Right.
That's that's true. So, like, when did you already decided that you want to come back to India, and but how did it go with the setting?
So I till my 4 or 5 years in my postdoc, I was not sure that I want to straight away want to go back to India, but
Mm,
I wanted to go for a
mm,
second postdoc in US.
mm, mm,
But I again, information is power, and at that time I was not having enough information. So
mm,
I then started to apply after 4 years of my postdoc and 5 years probably. And then I realized that most of the positions are actually entertaining people who are after 3 years of their postdocs,
mm,
if you want to apply for another postdoc. And also, they prefer people with, within 5 years of their PhD, after PhD. So
mm,
I was on the
mm,
borderline and I was not able to. And many people ask me a question, why you want to go for a second postdoc?
mhm,
And I was like that I want to get more experience or maybe want to go to US for a different kind of culture in science. But somehow then I decided, okay, maybe it's not worth. Let's maybe continue 1 or 2 more years here and do something more, and I will apply from here itself. And even my
mm,
mentor suggested me that why you want to go? You already are set here. You can do a lot. Why you want to move? And
mm,
at that time, I had my son who was born just that time, and he was very young.
mm,
So I thought maybe it's a hassle that to move with a small kid here to there and then to set up again. It might waste a lot of my time, and that was a correct decision actually. So I stayed there and start to apply from there itself after 6 years, and then ACTREC was my first application. And then
mm,
when I
and
got the call, I was kind of surprised. Oh my god!
mm,
And then I told my mentor, I was kind of a scared. Oh my god, it happened! Now they are calling me for interview. Then I
mm,
was very afraid that okay, but I'm one of my projects was in the kind of still not completed, was in kind of a
mm,
completion, and it was not
mm,
published yet. So I told her, see, this is happening, and I have to go for the interview. So my mentor thought, okay, maybe let her go, and so just a chance, right? She might not get. So why not to apply and go? Okay, you go. So I went. It was in 2018 November 28th, and I had my interview at that day. This was the first time I came to Mumbai. I was never been there before.
mm,
So, oh, and there was 28 candidate for that post, and we talked with all these candidates, and then I was not. My interview went well according to me, but I was still afraid that I don't know what will happen. So I came back
mm,
after my interview, and
mm.
then in January I got a letter from there that you have been selected, and at that time I literally cried. I was not sure, and again more afraid. Now I have to really go. And my project was also like in the last phase of building a paper. So
Mm,
then I
mm,
asked my mentor, okay, this happened, and she was like, oh my god, it's real now.
mm,
So she
mm.
said, you must complete this project and make some
Then
provision. So I worked even harder. We took an extension for 4 or 5 months, and I
mm,
completed the whole project there and then moved here.
that's really, so, like, the life moves ahead with the
Yeah.
science. So how did you find it, like, compared to Israel? Now you were back in India, but now with a kid and family and everything.
Right. So it, I was already mentally ready that what I'm going to face
Mm,
there, that I will have to make
mm,
my own lab, things will be
mm,
delayed. This is not the same system. I already knew that. It's not that I was not
mm,
aware,
mm,
but I was
mm,
too happy to do that. because this is what I
Start.
always dreamt of. That yes, I
Always
have to
dream.
be an independent mentor. I would have to
Mm,
have my own lab and do my own ideas there. So when I came back, I got a empty space where I have to start with the denting, painting,
mhm,
everything. So in the initial phases was quite slow, and then
mm.
after 6 months when I came here, COVID started. I joined
Uh,
in July 1st, 2019 here, and
mhm,
my lab was just
mhm,
ready to start. In 6 months, I took some trainees with me. We
mm,
were kind of ready to start with all my enthusiasm, and I got a very nice set of people in the beginning. There was so much happy enthusiasts. We were not having many things in the lab, still running here and there. Again, institutional culture, I like it very much. You go in
mm.
any lab, you stand in any lab, you can work. So nobody
Yeah.
was like stopped, even if we did not have all the equipment. But then suddenly COVID started in February,
Mm,
and everything was locked down. So whole 1 to 2 years we suffered because people were not allowed to enter the lab, only two people at a time in the lab. So it was a very, very, very challenging time. This 2 years
mm,
that we kind of suffer. Also, PhD students suffered a lot, isolations and all. So not a easy time actually after that. But all the time we were very much enthusiastic to work. This did not change even after all these hurdles. But yes, it could
mm,
have been smoother if this COVID thing would not be there, and things would have much more stabilized for us if this phase was not there. But yes, they did not made no did not make a huge difference to us because it was for everybody. This COVID was for everybody,
mm,
right? So it did not
mm,
stop us for doing what we wanted to do. Yes, it was slow, it was
mm,
stopped, it was in fragments, but yes, it was for everybody, right? We were
mm,
not alone there.
mm, mm. Right, right, right. COVID was a quite challenging time, especially for new PIs like you who joined
Definitely.
just
Yeah.
before that. Yeah. So, like, so, since you like mentioned about your journey and it was really fascinating and motivating, would you also like to tell us a little bit about your research now?
Sure. Yeah.
Maybe do you also have a presentation?
I have
I
a
guess.
presentation there. I can
Mm,
show you
great.
some things.
Yeah, yeah,
And
absolutely.
I can skip some slide because sometimes you need to show multiple things in order to make other people understand. But I will just be
Yeah,
quick
yeah,
with those slides. So it's like
yeah,
15
yeah,
minutes for this presenting time.
yeah, definitely.
Okay. So let me share this. So are you able to see the slide?
Yeah,
Okay. So this is our topic of day, and I would just like to go over briefly on certain things that we are trying to do.
sure.
So, I would briefly introduce what is triple negative breast cancer. So, as you see here, yeah. So, breast cancer, it's not full screen. Actually, it is full
Slideshow
screen.
mode.
Yeah, it is on slideshow, but sometimes it happens with
Ah,
the,
okay. Laptop.
it
Yeah,
is, yeah.
yeah.
Okay, maybe I share the whole screen, then it will be better.
That would be the best if you
Yeah,
can.
this sometimes it is stuck with these things. So now can you see the full screen?
Yes, yes. it's visible.
Yeah, great. So, breast cancer can originate from any tissues, either in lobules or the ducts. And And when you have this triple negative breast cancer, most of the time the origin is basal layer, which is not from the luminal layer. This is the luminal, which is actually having estrogen receptor, progesterone receptor positivity, or HER2 amplification. But this is most of the time is not originating from the lumen, but the basal layer, which is myoepithelial in nature. And this is why this cancer is very different from the other subtype of cancer, which is most of the time is detected in elderly women. And triple negative breast cancer is highly aggressive type of disease, and because it does not have estrogen, progesterone, HER2 targeted therapy, the only therapy that you can use is the chemotherapy, because the targets are not available. And because it's a myoepithelial type of thing, it proliferates very fast. It has a very high Ki67 score. So people classify this tumor into, when the tumor section comes and it is negative for ER/PR, but you can see how much Ki67 there is. So that means they are highly proliferative in nature. So this is how you classify them in the clinic. And the point is, they are more aggressive and they metastasize into very important organs like lung and brain. Other type of tumor which are non-TNBC, they goes to bone, liver, and other. But this is the lethal type of tumor which enters into liver, lung, and brain. And because of that, actually the mortality is very high. That means this tumor is highly aggressive in terms of dividing and in terms of metastasizing to very important organs. And this makes this tumor very much deleterious. You don't have therapy, you don't have. And another thing what happens here, when I was reading this literature, and I came to know that in western population the proportion of triple negative breast cancer is up to 20%, but in India you have much higher. In South Asian countries including India, the mean prevalence is 30%. That means every third woman who is detected by breast cancer would be triple negative breast cancer. So there might be many genetic and other factors that is making this kind of predisposition to TNBCs, but that means this disease is more relevant for Indian population, and we must study this. So, as I told you, the conventional treatment is chemotherapy, followed by surgery and radiotherapy, and there are very few targeted therapies which are available, which makes more difficulty to treat the disease. And here you can see, if we talk about survival of these patients, so PCR means you get complete pathological response, that the tumor are totally eradicated by the therapy. And if you have a residual disease, RD, which means that the tumor was not treated well with the therapy and still you have the tumor. So if you see that the non-TNBC disease, if the TNBC and non-TNBCs are treated well, so there's no much difference in TNBC versus non-TNBC, patient would survive. But if TNBC patient is not treated well, you have a residual disease, they will die much faster, and within 3 years, half of the population of the TNBC patient will die, which is very sad, because most of the TNBC patients are young women within 40-45 years, and they have small kids, right? So there's a huge difference between the survival when the TNBC is not treated well. So then we ask the question that how to increase the response to chemotherapy to this tumor, why people are not responding, why they are metastasizing, why they are dying, what we can actually do, how to increase the response to chemotherapy. And if people are not responding anymore to the chemotherapy, what alternative therapies can be provided to the people who are already chemoresistant? So these are the two questions that we are every time asking in the lab, to find out what we can do, how we can support this patient, and so on. So we are using multiple type of tools, techniques to address this question. We are doing in vitro systems, 2D cultures, 3D cultures, spheroid, organoids, multiple devices with collaborations. And we are also using mice models of xenografts, genetic model, PDX. So combining all these models, every model has their own power, their own limitations, and also equally applying in silico techniques like mathematical modeling, AI-based techniques to predict after you stain tumors and so on. So by integrating all these techniques and together, we are trying to address what is happening into the tumor when it is getting resistance, right? And then we came up with a concept of drug tolerance in this case. As you can see in this image, when the initial tumor, which is therapy naive, is treated with the chemotherapy, not every cell would respond, and there will be a lot of reprogramming within that when the drug is actually getting into the disease, and some of the tumor will become residual tumor with tolerance. And there could be proteomic changes, transcriptomic changes, metabolomic changes, and secretomic changes. And once the therapy is off, again the tumor will go and grow and as well as metastasize in this case. So why we wanted to have this model as a drug tolerant model? There are so many things that these TNBCs are quite heterogeneous. They have four subtypes. Most of the time in clinic, people have already understood that this is not a genetic mechanism. It's a non-genetic mechanism how this cell become resistant or tolerant to the chemotherapy. And most of the time, people have highlighted in the clinical data that there might be reprogramming. And not only that, they also change the state. They also become EMT, they metastasize much more once they become residual or drug tolerant. So then we came up with this, idea that this model in TNBC must be a drug tolerant model with all these features that it has. And then we are asking a question in that particular system itself, that to understand this thing, that why these cells are not responding, what is the duration of the therapy, what is the window of reprogramming. Even if they are reprogrammed now, they must have some vulnerabilities, right? It's not that the tumor is like good for everything. They will not die by any other manipulation. So we are also trying to understand at the same time what are this vulnerability, how we can kill this drug tolerant and metastatic tumors in triple negative breast cancer. So for that, we came up with a model that we chase the cell line with long term as well as short term with the drug, and we identify certain phases. That there's a initial phase of chemo tolerant. It's a drug cell death phase by the drug. Then start to these are the tolerant cell which will no more die even if you treat them again and again. This is a stabilized tolerant phase. Then there's a recovery phase when the drug is off. Then there's a proliferative phase. So the cell again start to proliferate. We are looking into this longitudinal window in terms of within like 20 days and 21 days or 1 month, and also in the short time of window when there's a cytotoxic effect, and then there's a phase where no more cells are dying. So we are also zooming in into that phase. And then we are trying to characterize, and we identify two type of population. One is dormant drug tolerant population, which is happening up to this time when the cells are no more dividing. And then they are proliferating, which is called proliferating drug tolerant persisters, which is here. They are drug tolerant, they are also proliferating. So we identify these two population and multiple type of TNBC cells, and we then characterized. And then we are trying to ask question with these two types of dormant as well as proliferating cells, that with multiple modeling in vitro and in vivo. We are asking some immunotherapeutic question, that some molecules actually increase their like CD1D. We are trying to understand family of BET, bromodomain proteins, because these are highly expressed and also now more activated in drug tolerant cells. What is their role? Because the drugs are available. We're asking EMT type of question, why the cells are changing their phase, if this phase is stable or it is in the middle. And we found that there are some partial EMTs, full EMTs, non-reversible EMTs in different subtype of tumors. We are more a lot of focus is towards the iron metabolism and ferroptosis, because we know that these triple negative cancers are very special when we talk about ferroptosis, how this ferroptosis pathway is actually altered. And this has been shown with multiple drug tolerant cells in other tumors like lung and colorectal. TNBCs are highly secretory in nature. So how the secretome actually influence other cells, stromal cells, or even crosstalk between the cells, this is something also we are focused on. Receptor tyrosine kinases and receptors are one of my favorite things that I have been involved starting from my PhD self, and several receptor like EGFR, which is expressed in half of the population of TNBC, highly expressed, and their partners. We are looking into that for a drug tolerance particular thing, and we are trying to understand what is happening in this population and how we can treat that. So we characterize those, and we try to see they are tolerant, they are still resistant even after they proliferate, and they can form more colonies, and the tumors are growing larger when they have drug tolerance cell implanted in the mice. They have high Ki-67 scores. And then the asked question is why they are actually so aggressive, why these DTP drug tolerant persisters are so aggressive, what is happening there. And then the second question is how we can eliminate these cells if they are. So in order to ask this question, we simply looked that what is the signaling, what is the tyrosine mark and other phosphoserine marks in this type of lysates where you have drug tolerant persisters. So we took paclitaxel, bractoctal, and persistent, and can just probe with the phosphotyrosine antibody. Also, phosphocyrine we have done. So the signal is very high into these drug tolerance, and as you can see, at least 3 to 4 fold more higher in general. And then we try to look at the signaling, and you can see that all the RTK signaling is very high into these cells when they are tolerant. RNA-seq also suggests the same, that the PI3 kinase and ERK signaling is very high into these cells. So then that means they are also building on the signaling. That means the higher the RTK downstream signaling is, they can proliferate. This is actually supporting the cell survival and so on. Then we did mass spec analysis, and surprisingly, we see that EGFR, which is highly expressed, it is now very less expressed in terms of protein amount in mass spec data. And then we try to see in our Western blot and see, oh, yes, it is highly down regulated in the persister cell. But surprisingly, the phosphoform of this receptor is now much more phosphorated, is much more active. Even you can see the protein and the amount of the phosphorylation that it has in multiple subtypes of TNBCs. And we can see up to 55 folds of higher phosphorylation of this receptor, despite this receptor is so less expressed into these cells. So then we try to investigate it, why it is happening, what is going on with this receptor. So we could see that the localization of this receptor actually has changed in persister cells, drug tolerant cells. You can see in naive cells in MD468 BL1 type. So the membrane localization is there in the receptor. But when you see that in the persister paclitaxel, doxorubicin, in these two different subtypes of cells, the localization from membrane to cytosol, it is changed. You see that there's a puncta structure there also in some tolerant cells. Here also the receptor is from the membrane. It is in the membrane, now it is changed in the cytosol. So it wanted to know what is happening. So then we also took the other data of survival, and everything was matching that low EGFR receptor is actually bad for patients which are treated with chemotherapy. So it actually aligns to what we know. And also in patient data, in metastatic, you have less and less of EGFR expression in TNBC. Lot of the data in survival plots in patients with chemotherapy suggests, yes, if you have high expression of EGFR, the survival is good for the TNBC patient. If you have low, then the patients are actually dying. This is clinical data. This suggests, yes, there's a significance of this finding. Same goes for the cohort which are treated with doxotaxel, and patient who are doing good have higher amount of EGFR. Patient who are not doing good with the therapy, they have lower amount of EGFR. So everything is aligned to what we know. Then we try to stain further, and we see that this EGFR actually is degrading, and you can recover by chloroquine treatment. So EGFR is actually high, maybe expression is good, but because of the activation is very high, it is actually very fast degrading into the persister cells, drug tolerant cell, and it is happening almost all the cell line that you can see. You can recover this receptor in the puncta structure. That means the receptor has totally changed its path. It is highly phosphorylated, highly degraded, but then also we can see in the Western blot, yes, it is recovered by chloroquine. That means it is degrading. So one of the factor which is called NDRG1, which is related to NMIG down-regulated protein 1, is actually a protein that can facilitate the degradation of this protein, and it's a protein that is upregulated when the cells are under stress conditions. So this molecule NDRG1 is highly expressed in persister cells, and because of that, actually, it's also co-localized to the EGFR receptor, and this is making it degrade. And this also has a relation with patient data. So then we ask, okay, but EGFR is degrading, but still the signaling is very high, the RTK signaling is high. What is happening? So we could see there's a switch to EGFR to SRC family kinases in this drug tolerant cells. SRC and lynfin kinases are highly expressed into these DTP persister populations, and these are actually becoming the hub of the RTK downstream signaling. And we could see that these are highly expressed in multiple type of cells when they become persisters. So also at the same time, we looked into some literature, and we could see, yes, this is a PDX model study from drug tolerant patients, and they could see that EGFR, SRC, and filin kinase are highly expressed into these PDX models. So this is kind of a proof of concept. What we are doing is true and also aligns to the patients. And then we try to see, okay, can we suppress the signaling? We took paclitaxel, and we also took EGFR RTK inhibitors, and could see that, yes, these tumors are actually responding to EGFR suppression because it's very high. And also with paclitaxel, you can suppress these tumors in 3D condition, 2D condition. You can actually use EGFR drugs with paclitaxel if this kind of things are happening. So we just validated the effect of EGFR suppression to the signaling, and yes, it was actually suppressing downstream signaling. So this is one study that we have done, that the SARC lymphin kinases are becoming hub with EGFR, although EGFR is degraded, but these are the kinases which are actually switching over and then carrying all the RTK function. And when you suppress EGFR, somehow their activation is downregulated, and you can suppress the tumor which is happening there after tolerance. So another study, we try to understand that what are the vulnerabilities of this tumor, and there are screens that we identify that triple negative breast cancer can be killed by ferroptosis, and this was a combination therapy with BET protein and proteasomal inhibitor proteasome. We can see that yes, it is actually affected, and we also relate with clinical data here that yes, it is relevant whatever found in the screen. So this was the combination that we were focused on: bromodomain inhibitors and proteasome, which is an proteasomal drug. So that induces ferroptosis in most of the TNBCs that we have tested. These are all the cell lines. This is the cell death in yellow. So because this combination can downregulate GPX4, which is a central enzyme for glutathione conversion into reduced glutathione. So that actually is very lethal for the this combination is lethal in triple negative breast cancer. And then we are try to understand why it is happening, what is the redox valence, and so on. So then we understand that there are so many genes, like 700 genes, which are actually in the pathway of ferroptosis, glutathione metabolism, iron metabolism. They are very differentially expressed in triple negative breast cancer or basal like tumors in the patient data. As you can see, this cluster is completely different from rest of the breast cancer. These are the 700 genes. That means they are totally molecularly different, this disease. And these are the maybe vulnerabilities of this tumor. We can hit there. And we also validated everything in the patient samples in ISC conditions, that these receptors, iron metabolism is actually differentially regulated. And the cells of TNBCs are highly susceptible for the induction of ferroptosis, which is an iron-mediated cell death. As you can see, all the TNBC would die, non-TNBC will not die if you try to induce ferroptosis. There are molecules which are regulators of ferroptosis, which they are differentially expressed between TNBC and non-TNBCs. Same happens in the patient. Another protein which I'm talking about, BRD4. Why BRD4 is actually inducing ferroptosis? It is highly expressed in triple negative cell lines. This is the CCLA data as compared to non-TNBCs. You can see that how much it is high. And then same thing happens in the patients also. So we are trying to understand if GPX4 enzyme or ferroptosis vulnerability can also be a vulnerability of the drug tolerant cells, although TNBCs are still vulnerable to ferroptosis. So then we look it into the clinical data. Yes, GPX4 is highly suppressed there. They might in the patient data itself. And high expression of GPX4 actually supported if the patients are treated with chemotherapy. This is what we found. And we could see that there are markers of EMT which are exclusively not present if GPX4 is present. So GPX4 is oppositely correlated to EMT marker. So we identified the role of GPX4 in EMT. In tumor, they are exclusively present. If GPX4 is present, PIMNT, which is a marker of EMT, is not present. So this also happens in the tumor. So we understand an opposite correlation between EMT marker and GPX4 protein. That means it is also initiating some EMT program if it is not present. So there are some other things that these are just the data that suggest that lipid peroxidation is very high in the persister cells, and we can actually kill those with the glutathione inhibition. So these cells are actually vulnerable to that. And another vulnerability we identified that these cells are dependent on autophagy, and then if you suppress autophagy by either chloroquine or by this another drug, these cells can die, so they cannot live without autophagy. And so this is something that I would like to say my journey, that this is where in 2008 I was presenting a poster in my PhD in Bose Institute. This is how I used to look. Then in Weizmann, starting from 2012, I used to work as a postdoc. This is my lab when I entered, at Trek in 2019. This was my empty lab after painting and all. And then in 2020, we started my group with most of them were trainees, only one PhD student. And then we were so happy to, answer every question and whatever we could do at that time. So I've been associated with many talented, enthusiastic people. And this is the pictures over time that we had many people and enjoyed several moments together. And then also my groups which are changing, like many trainees are getting trained. Up till now I have trained maybe 30 master students, over 30 master students, and many others. And this is my current group, where I have 4 PhD students and some are project student masters and all. And these are my collaborators. This is from Wiseman, Professor Strutman. So he was the one that showed me how to do organoids for or tumoroids for different type of
Mm.
tumors. He's my Itan Rupen is my collaborator for systems biology. He's my collaborator from IITR for metabolism. Dr. Sora from my own institute. Mohit Kumar Jolly for many bioinformatic analysis, and Dr. Flavio, with whom we got a international grant, Indo-French grant, and he has having very nice mice, genetic mice, which only generate, triple negative breast cancer. So with them, we are
Mm.
conducting some ferroptosis kind of studies. And our lab
Mm-hmm.
is generously funded by DAE, SERV, and as well as CEFIPRA. So with this, I would like to thank you all, and would like
Good.
to address some questions.
That was a uh, really good presentation with really targeted therapy and starting from basics and how cancer is made, how cancer tumors are formed. So I would say that overall, I'm sure that people would have understood many things, but also many things were very new, like ferroptosis and everything, because not many people, when I entered the field of cell death, I came to know that there are more than one or two types of cell death which we don't know,
Hmm.
but now it's so
Now there are over
common.
18
So
type of cell death.
yes, and if you say something like this, I mean, if you say this for for them who don't know types of cell death, then you need to explain them. Okay, there is a difference between
Right,
a pyroptosis and necroptosis.
right. Many new T
So
cells come in.
pyroptosis, yeah, because like one of our collaborator here, we're meeting them tomorrow, Marcus Conrad. He also works on pyroptosis. So
He's
that was
also kind
really,
of a collaborator of us. He's doing fantastic job in this
yeah.
process field. Marcus is
Yeah,
great
Marcus
actually.
Marcus is really,
Yeah,
he's
he's
a
one of
he's
the
a good
pioneer
PI.
in ferroptosis,
True.
studies.
Yeah, true. So he only introduced me to the field of ferroprocess because I wasn't knowing anything about it when I entered my PhD. But I went to a retreat, and then he was taking our lectures there for ferroprocess. So it's quite
Right.
good. So there are few questions today on the entire topic, and one of the question in relation to your maybe inos project that somebody asked was that how does inos mediated inflammation influence the regulation, especially in the case of ferroptosis in physiological condition, especially because I mean, there are very less known facts about ferroptosis in physiological conditions. So
Right. So we haven't explored by ourselves the role of iron-OS in
mm-hmm,
ferroptosis, but I'm very sure
mm-hmm,
it must be there because whenever we talk about ROS, inos
mm-hmm,
is an enzyme that actually
yeah,
increases cell ROS, and this is a
yeah,
specifically induced type of ROS whenever
yeah,
any
yeah,
inflammatory pathway is on. This is a byproduct of
yeah,
any inflammatory condition, and people have already associated inflammation with ferroptosis. There are pretty good studies which suggest that the link, but maybe it's not very clear, or maybe I'm not aware of this literature. Probably it's a high time I should look into NOS and connection of lipid peroxidation in cancer cells. So, but inflammation, it is known probably that high ROS, and the point is ferroptosis will not take place unless you have high iron intake, and you need a
yeah,
separate set of receptors to do that. The intake of iron has to be increased by transferrin receptor and ferritin
yeah,
proteins, and the out the transport of iron outside in from the cell should also be stopped at the same time. And then
yeah.
if you have ROS in this condition being iron high in the cells, that
Mm,
provokes the ferroptosis and phantom reaction, and then the membranes are damaged
mm,
because of the free radicals generated there. So INOS might contribute. I may not have read a specific literature about INOS, but I
Mm.
think I should look into that. That's a very good idea for me.
Yeah.
Fair
So
possibility. But
mm, so I also had one question. Maybe I was also not much informed, but like EGFR, the pathway that you showed had direct correlation with NF kappa B targets. So did
yeah.
you ever check on that? What is the effect, and if there is an activation of NF kappa B, which I assume is active in cancer due to survival. So
Right.
if NF kappa B signaling is active, do you have you ever found any direct or indirect link to ferroptosis?
So NF-kappa B is a very central molecule which will be activated by any kind of cytokine, growth factor, and
Yeah.
even stress type of
Mm.
signals, right?
Yeah.
And people have
Yeah.
already found that NF-kappa B is so central to even normal cells that you can't target
Hmm.
that. It's toxic.
Yeah.
So this is
Yeah.
another problem with NF-kappa B. So initially people were very enthusiastic about NF-kappa B and there were several trials. It was long back
Mm.
in like maybe 20 years ago or something.
Mm.
And most of
Mm.
the trial failed because of the toxicity. So
Yeah.
even if
Yeah.
you find that, I'm sure NF-kappa B would be also regulated by EGFR also because it's an ATK dependent pathway there, but
Yeah.
not
Yeah.
sure that how you can target NF-kappa B because again the toxicity is an issue and people have
Mm.
probably gave up on this kind of trials when they want to target
Mm-hmm.
NF-kappa B as such, because then you have immunosuppression, you have toxicity, many other issues crop up. And
Yeah.
this is why I particularly not looking into NF-kappa B because the rationale is not there. Even if it is there, there are some molecules like which are central but also important for the other type of cells, normal
Yeah.
cells.
Mm.
So you can't actually target them. And same problem is also there with GPX4 also, because
Yeah.
GPX4 is also very central to brain cell, kidney cells. So if you suppress GPX4, there's a lot of toxicity in the normal tissues also. But you
Mm.
want to look for a threshold value and as well as a combination. Where what you
Mm.
want to combine with GPX4 that makes a synthetic lethality kind of concept in
Mm.
the cancer
Mm.
cell, which is specific to cancer. So there are many molecules which are very central, but you can't target them. This is one of the problem. And I'm sure NF-kappa B being also generator of the ROS because there are so many cytokines which
Mm.
are dependent on NF-kappa B. And again, they
Yeah.
will generate
Yeah.
ROS. NOS is again will be there, right?
And
It's a
then
loop.
yeah,
So maybe you
it's
can
a
crack
loop.
it, but you need to again find a kind of a threshold between cancer and
Mm.
normal cells. Only then I
Mm.
think these things are applicable at all.
Mm. Mm. Yeah, that's you cannot completely inhibit
Right.
otherwise.
We have looked
Yeah.
because we bought an antibody and it was activated, but then we did not bother ourselves too much.
But the antibody is
Because
not that
yeah,
great.
because NF kappa is something which I know from my PhD time. So, but I know what is the limitation with that also.
Yeah, yeah, great. So, like considering, I mean, not just India, but all over the world, many places, insurance companies, are correlated with the clinical trials and the targeted therapies. So, like, if you deal with them or have you ever deal with them, then like what are their takes for the treatment with the combination drugs nowadays? Because like there were many drugs which are not uh, I would say sensitive towards cancer. So like where is the world shifting today for the targeted therapy?
So most of the time, whenever there's a discovery, like there's a whole set of things that happens in the translational research.
Hmm.
So initially you
Hmm.
have a discovery, then you have a preclinical mice model. You prove that yes, this therapy is working. And there's a lot of things that happening at the end of pharmacokinetics. That how this drug is actually absorbed, how is the excretion of the drug, how much retention
Hmm.
time. So
Hmm.
how is the formulation, so then a drug actually, is marketed after all these things are passed. That there's a
Hmm.
clinical data that suggests pharmacokinetics, toxicity level has been studied. So phase one trial starts with this thing that you have a drug in the market, which is known for certain activity, toxicity, pharmacokinetics, stabilized formulation, and so on. So the
Hmm.
companies have an interest that they want to put their drugs forward for the later on for the approval that these are the drugs
mm,
that can be given to the patient. For that, they need to first pass 3 phase clinical trials, 1 to 3, and for
mm,
that they have to sponsor certain patients for the clinical trials
mm,
without asking money. Otherwise, they will never be passed for commercial use, right? So the first clinical trial, maybe in a set of patients, the companies come up and they have to convince the doctors and the people who are involved in the trials, and enrollment is
mm,
done for safety studies only, and they may be up to 50 patients or even healthy individuals. That the
mm,
first priority is the safety of the drug. Whether the drug is not doing, it should, maybe it's not doing the job, but it should not do something else which is adverse.
mm.
It is okay if the drug is not working in the human for what it is supposed to do, but it should not do deleterious effect. This is
Toxic.
the first phase objective. So if it,
Mm,
the drug passes the first phase, that it is safe to use, there's no toxicity involved, it is tolerable, it is excreted out of the cell, of the body
mm,
safely, and everything is managed, you don't have much side
mm,
effect. So then it goes to the second phase of clinical trial. Of course, the company
mm,
has to bear the cost and so on, and you need to tell
mm,
people who are suffering. For suppose it's an anti-cancer drug, the people have to
mm,
agree to be enrolled into that trial, and they will be informed for everything that you might have a good effect,
mm,
but it is not guaranteed. You might
mm,
have some adverse effect because now you're a cancer patient, not a healthy individual actually. You might have some vomiting, some headache, and maybe other things are involved with this drug because you have been treated with chemotherapy. So they are informed, they agree to that, and then the second cohort starts for the phase 2 clinical trial, where the objective is whether the drug is working for what is supposed to do. And
mm,
many parameters are actually tightly regulated and also tightly evaluated to infer that studies whether this can
mm,
go to third clinical trial where larger population
mm,
has to be studied with the same effect. So if the drug has
mm,
some good effect, that yes, some at least 50% or maybe 40% people were benefited, maybe 10%, 20% were completely cured with this drug. So then
mm,
there's a decision, yes, it is safe to use, less toxic, tolerably
mhm,
toxic, and you have a
mm,
marginal effect or maybe good effect when whether this drug can pass to the third phase of trial, where maybe
mm,
200, 300 patient will be treated with drug, and then only you
mm,
will have an approval. Yes, those patients had a good statistic. There's the overall
mm,
survival, some this distance metastasis free survival, or some survival benefits are there over the people who are not treated in the other arm without this drug.
mm,
So then only the FDA approval or Indian regulatory bodies approval is in place for the commercial use. So in
mm,
that case, they have to go a long way from the discovery to the clinical trials. And for some drugs, it is quite fast because
mm,
maybe the population is waiting for that drug. Like you can have an example of COVID vaccines. So it was
mm.
pretty fast because all the
Yeah,
population actually was waiting for this thing to happen. So people
yeah,
can
yeah,
push it where you don't have to wait for a longer time point study.
yeah.
You have to make conclusion. But for cancer, people wait till maybe 4 to 5 years to get the result of this first
Mm,
phase trials, and then only the approval
mm,
is in place, and then the drugs is commercialized to the people. And then
mm,
also
mm,
it is given to a very special set where they can have benefit, not to everybody. Like herceptin, which is
mm,
the HER2 drug, it should be only given if the patients are found highly positive for HER2 expression in the tumor, right?
Not true.
Because
Mm,
it would be toxic otherwise, and also it is also costly. Why people have to
mm,
suffer for nothing if they are not benefited? So the
true.
criteria to
Mm,
whom you can give that drug is also very stringent.
mm, mm. True, true. That's certainly true. It was a really nice description about the phases of clinical trials. So we have few questions, ma'am. Do you have some more
Sure,
5 minutes so we can?
sure. I'll be
So
happy to
there
answer.
was sure. So there is a question from Mr. Devanath. That what's the present advancement to early detection and I would rather go with detection right now for the TNBC patients.
So, I would say in general, awareness towards cancer has been increased a lot from past 10 years or maybe 20 years, because
Mm,
females are more educated. They can have their self
mm,
detection also. They can approach to the clinic, and the health
mm,
worker are also actually going and educating people much more. And that we have seen in the number of patients detected every year, that has been increased dramatically. And people say worldwide, every eighth woman is actually affected by breast cancer in her lifetime. So it's a lot, right?
mm, mm,
So you
mm.
become so fearful that, oh my God, like every eighth woman, right? So
Yeah.
this is because not only that the disease is happening more into the population, but also the detection system is now more
Mm,
robust. People are
mm,
more aware. They approach to the clinic once they detect any type of nodule in their breast, or they feel something painful or something. So they reach to the clinic. They know it could be a breast cancer, and breast cancer is treatable. People also
mm,
understand nowadays. So they have a positive outlook. Yes, go early. And we are also doing a lot of campaign that early detection, and this are this every year we run many programs for that. We outreach to villages. We have teams. So people are actually aware of that nowadays, and they are not hesitant to reach to the clinic, and they also not very afraid, because 75% breast cancer is ER/PR positive, that happens into the later ages after menopause, and this is highly treatable because of the targeted drugs available. And I have
mm,
seen within my family people who were suffering, they have already 15 years of survival after getting treatment. 15 to 20 years survival after treatment is a, people are seeing that in ERPR positive. So there is a positivity about the treatment, that yes, you get treated, you will be surviving very good, and the treatment is also very nice. So things are moving to a positive direction. It's not that people are more
mm,
fearful about cancer nowadays, because better treatment is available, and I think many good doctors and highly educated people who can screen and detect, they are available
mm.
here.
That's certainly true. So there was one more question from Simran. So she's asking that what are your thoughts on plant-based, plant-derived exosomes carrying secondary metabolites on triple negative breast cancer, and how can you investigate their apoptosis pathway, rather I would say cell death
Cell death
pathways, program
pathways.
cell death. Yeah.
So it's not new that many drugs, especially chemotherapy drugs, are derivative of plant compound. Paclitaxel is one of the
Mm-hmm.
top example there, and they're highly effective. So people are exploring plant-based medicines, and they are, and these are most of the time these metabolites. They are cyclic
Mm-hmm.
compounds and others which have been, tested in animal tissues for their effects. Many of them are anti-inflammatory. Many of them are anti-cancer. And there are many other effects. Some are protective, cytoprotective. So there are many studies which are going on here, and we actually ECTREG is actually establishing another center for that, which is called Khapoli Center ECTREG, which is mostly focused on the plants, and they will grow those medicinal value plants. They will extract them, they will test them, and try to integrate this set of medicine also to the mainstream. So there are
Mm-hmm.
things which are happening, and we are also testing certain drugs with collaboration. We have a lot of drug libraries from plant-based compound
Mm-hmm.
in India.
Mm-hmm.
Even in our extract, there are some people like Dr. Vikram Gota. He's a pharmacologist, so he has a whole set of library of compounds. We are testing that in drug
Mm-hmm.
resistant or drug tolerant triple negative breast cancers. So in combination with other targeted therapy. So yes, there is a set of success that we see, that yes, they are effective, and the extraction
Mm-hmm.
method and many things are different, that you want to extract a set of compound or so on. And some of them are actually can be conjugated to target organelles like mitochondria. So a lot of
Mm-hmm.
research is happening here, even in our center and worldwide also, that people are
Mm-hmm.
targeting many type of cancer with plant-based drugs. And it was a huge success in the past, when we already learned from the past that yes, these are effective molecules, but we just need to look into a particular scenario and try to
Mm-hmm.
conjugate or try to somehow encapsulate them in a better way, so that targeting is better, and try to find out what can be given together. Sometimes even the opposite is also correct, that people are taking some herbal drugs, some formulations, and because of them the toxicity to chemotherapy has been increased. Some cases of cell means death of patient also happened, and doctors actually always asking them whether you are taking any other drug with this drug should be stopped first. Don't try to mix
Mm.
many type of drugs together with chemotherapy. It can have a very toxic effect and we will not be able to know why it is happening. So the
Mm.
opposite is also true, but again, knowledge is a tool where you can have an experimental evidence that yes, these things can
Evidence.
work together, can be given together.
Mm.
And because of that, the new center is actually established, getting to be established soon, that would very systematically test these kind of approaches, these plant medicines, that how they are effective. And we are testing some of them in triple negative breast cancer also.
Mm. Yeah, plant-based, I would say strategy is quite evolved in India, but yeah, the main thing that, as you said, the how you encapsulate it
Yeah.
and like where do you
Rationalize.
use it
Yep.
is
Yeah.
really important. Yeah, it could have multiple targets at
Yeah.
different places. Great. So, like usually, I ask one last question to each and every PI or a scientist that we host. And the question goes like that: what is your favorite discovery? So, till now in science. So, there are many sets of answers that PIs give. I can give you some uh, example. So one of the PI came over and she said about the discovery of H plus and Cl minus ions. Then the discovery of qPCR, through which we are doing so much things, genotyping and everything evolved. So like what as per you is the discovery you feel is very important in science?
In science in general or biology, that matters.
Mm-hmm.
But I will say the discovery of light, that how much different nanometer wavelength the light has and how we can use that actually transform the whole set of things that we can look, how we can
Yeah.
visualize things happening. This is something that I always, you know I'm always, this is very close to my heart, that laser light, spectrophotometer light, UV light, X-ray light. People now understand there's so many different type of wavelengths that we can utilize just to look things. Otherwise, we can never look into that. When you
Mm.
see, this is a thing that you can believe. So all this repertoire of wavelengths that is enabling us to see different level of things, like discovery of X-ray. We could never see what is inside our body if the X-ray was not discovered, was not utilized by serendipity. That yes, you can see
Mm.
bones, you can see internal organs. The discovery of
Mm.
even spectrophotometers, that
Mm.
how we can see the wavelength changes when the molecular reactions are happening, how we can utilize
Mm.
laser light to see confocal microscopy, when every time we can zoom inside the cell and can see what is happening, what is moving, and even
Mm.
in the live cell setting. So this amazes me so much. When I see something, I believe, and when
I
I
can
can
understand.
chase things in a live scenario, I know what is happening. Then I can predict,
Mm.
I can form an experiment, and I
Mm.
know this much is possible. So that fascinates me to something that I can't even explain how much
Yeah,
fascinating it is.
yeah, absolutely. And to my notice, I mean, this is one of my research project as well for my PhD. There is a light receptor on the cancer cells which acts like TNF receptor. Probably, we don't know. We are looking into it, and it activates inflammation and cell death pathway. Just the
One of
interaction with the light.
Yeah, it's very true. One of the lecture from BRC, there was one very eminent scientist came to us, and he even had trials kind of there. If you are exposed to early morning light, if you're exposed to the midday light, and if you're exposed to the evening light, they all have different wavelengths, and the effect
Yeah,
is
yeah,
completely different on your body. Every cell can
yeah,
sense the light that is exposed to that cell,
yeah,
and some
yeah,
of them
yeah.
are pro-inflammatory, some of them are anti-inflammatory in nature. So this is why in our shastras people say early morning light is the most
Mm,
best light that you can
that's
absorb
light.
into your
Mm.
body, because the
Yeah.
wavelengths are different, the effect is different. It's a much more healthier light. But blue light also can do wonders. That is in the
Mm.
most of the time it's towards the evening when you have
Mm.
enrichment of blue wavelengths. So
Yeah.
yes, it is there, and people are actually looking into that. And whatever I hear about all these things, that makes me more, uh, love make me love science much more. That see, so many things are possible. We know so
Yeah.
less. We need to know
Yeah.
more,
Certainly.
and we try to think in an integrated way.
Yeah, certainly. So we are towards the end of this event, and it was really, really great to have you, ma'am. It was really insightful talk today. A bit longer, we extended a bit because it was quite interesting for everybody, and we had a lot of questions as well. So thank you so much for coming, and for the audience, we would like to deliver that. Please fill the feedback form. It would really help us, in the long term to invite other PIs, and also, your suggestions would matter a lot. That how could we improve next time or something like that. So I would like to please request audience, we would paste the feedback form in the YouTube link. It's already there, so please do fill up. And I would
Can
like
I
to
also
ask
feel this?
Somilia Mishra. Yeah, I would like to ask Somilia Mishra to please deliver vote of thanks today. He is also a PhD student at
Thanks, Mamita.
BARC, Mumbai.
Great. Yeah.
Yes. Yeah, that's also
Thank you
important.
so
Thank you so
much.
much,
And I
ma'am. Thank you so
really
much for
like
coming.
this session, and it's so good to talk to young people like when you are doing a great job for the, scientific society. A lot of congratulations to your whole team. Thank you
You,
very much.
ma'am,
And I
as
wish
well.
you
Thank you
all
so much
lot of
for
success.
coming.
Yeah, lot of
I
success
wish
to
that you
everybody who
could.
is working into this field. Lot of success.
Thank you, ma'am. I also wish you all the best for your research, and also I wish that you would uh, get a drug approved,
We
hopefully
are
near
looking into
future
that.
with
Yeah,
collaboration.
sure.
Good.
Thank you so
Thank you,
much.
ma'am.
Bye bye. Take
Bye
care.
bye.